Welcome to my video on the endomembrane system, a complex system found in eukaryotic cells. This system consists of organelles that are functionally related and connected by phospholipid membranes. This doesn't mean that they are physically connected, however, as organelles can also be connected via transport vesicles. It is found in every eukaryotic cell and consists of all cell organelles except the mitochondria and chloroplast. It plays a substantial role in protein synthesis and transportation of molecules. Structures of particular importance include the plasma membrane, the endoplasmic reticulum, the Golgi apparatus, and vesicles. Join me as we discover the structure and function of some of these components. First, let's start with the plasma membrane. This surrounds the cell, and as with all cell membranes, the plasma membrane consists of a phospholipid bilayer with some cholesterol, glycolipids, and various proteins embedded in it. Phospholipids are amphipathic molecules and have a hydrophilic head and hydrophobic tail. Therefore, it is semi-permeable and only lets certain molecules in and out. Next is the endoplasmic reticulum, which is usually the largest organelle in the cell and is continuous with the nuclear envelope. It can be divided into the rough EOR and the smooth EOR. The rough endoplasmic reticulum is studded with ribosomes, which have a major role in protein synthesis. Its other functions include lipid and protein transport. Indicated by its name, the smooth endoplasmic reticulum lacks ribosomes, and its functions include producing lipids and steroids. Another structure is the Golgi apparatus, and this is composed of cisternae. It is known by many as the sorting center of the cell, as it receives, sorts, stores, and distributes molecules in the cell. The cis side, the side closest to the endoplasmic reticulum, is the receiving end, where molecules enter, and then, after progressing through the structure and undergoing modification, molecules leave via the trans side. Finally, vesicles are other important structures in this system. They are small membrane-bound sacs with a short lifespan. They are formed when a segment of an already existing membrane pinches off, forming this independent structure. There are many types, including lysosomes, perioxysomes, endosomes, transport, and secretory vesicles. Their functions vary from degrading molecules to transporting them. For the final section of this video, we will learn about the formation of a vesicle and vesicular transport, as this is one of the main functions of the endomembrane system. This involves the budding of a vesicle from one organelle, known as the parent organelle, and then the transport of molecules to the target organelle, where fusion will occur. The first step involves selecting the correct molecules for transportation, and so the cargo bundles into a specific area on the membrane and forms a bulge, which then grows and becomes a bud. Vesicles usually bud from coated regions on the membrane to form coated vesicles. There are three types of coated vesicles: clathrin-coated vesicles, COP1, and COP2. Each have a different pathway of transportation in the cell. Cision proteins are then responsible for releasing this bud and forming the transport vesicle. This then travels to the target organelle by interacting with microtubules and actin filaments. Rab proteins and snares ensure fusion with the correct target membrane. V snares are found on the vesicle and T snares on the target membrane. Vesicles can then release their contents into the target organelle. That brings us to the end. Thank you for listening.